the various reasons that the 40N consortium is working so hard. And thus, CRISPR has come along as an ideal tool to perturb regulatory elements in their native genomic context. Of course, as a genome engineering tool, it works very well to test elements in their genomic context. Additionally, CRISPR, though it's not the first genome engineering tool, is massively parallelizable. So an entire pool of perturbations can be generated as CRISPR was standardly used with guide RNA target sequence that can direct your nuclease anywhere around the genome. A double-stranded break is created, and usually results in a small, about 10 base pair insertion or deletion. But there's also a version where the Cas9 nuclease is rendered enzymatically inactive. So it's nuclease dead Cas9 that's been tethered to the Crab repressor that induces heterochromatin approximately within 1 to 2 kb, and this has been shown to silence enhancers successfully. So in the past few years, single gene CRISPR deletion scans have come along, and these all work in a very similar way. Guide RNAs are scanned around usually a single locus. One guide RNA is delivered per cell, and then the cells are separated based on low versus high expression of a target gene. Thus, it's easy to try and associate induced perturbation with changes in the gene's expression. And these are great because you can test many non-coding perturbations in a pool all at once. But of course, they're only studying these candidate enhancers against one gene at a time, usually, or perhaps only a couple genes that are in the same pathway, something like that. So even though they're so high throughput, they're still limited in throughput because of the single gene phenotyping scheme. So ideally, we would be testing every candidate enhancer perturbation against many genes at a time, and ideally any gene that's expressed in the cell. And that was our direct motivation for what we originally called method that we originally called CRISPR QTL mapping. It's essentially you can refer to as multiplexed whole genome or whole transcriptome enhancer gene pair screen, and I'll tell you about that today. Okay, so we were for this we were directly inspired by human expression quantitative trait loci mapping. It's a high throughput method to test all genotype variants against all expressed genes captured in RNA-seq. How these usually work is that a population of people are genotyped for variants, and then each person in the population can be a data point, and if stratified by their genotype at any particular locus, the genotype can be correlated against all expressed genes that were captured in the RNA-seq data from tissues from those people. And if you see a correlation of a genotype at a locus with changes in the gene's expression, you can say that, okay, putatively in this variance region, there is a variant that is impacting expression of that target gene. And these are great because you can resort the population and you reuse the data set over and over again to test all variants you've captured against all transcribed genes you've captured. However, as an enhancer gene pair screen, EQTL studies are limited by linkage disequilibrium because variants fall long haplotype blocks, and it's very difficult to figure out which is the causal gene that's driving changes in the gene's expression. So we wanted to take a step back and ask if we could apply this logic to the CRISPR pooled screen framework that I just started by telling you about. So in this, instead of people, we would be talking about cells in a population. And instead of naturally occurring genetic variants, we would be talking about large effect CRISPR perturbations distributed all around the genome. And the idea then is that we can stratify the population of cells based on any one guide RNA or perturbation, and then try to correlate against all possible transcripts captured within a cell to be able to tie that candidate enhancer's perturbation with the correlated changes in a gene's expression, and thus nominate an enhancer gene pair. And then we can use the same logic of eQTL mapping to stratify the population over and over again based on every guide RNA and try and test every candidate enhancer's perturbation against changes in any, putatively any gene's expression. So one way to do this, of course, would be to create monoclonal lines and perform bulk RNA-seq on those populations, just like in actual human eQTL studies. But fortunately, single-cell RNA-seq has come along that enables us to deliver perturbations per cell, and then each cell can be a data point because you can capture nearly the whole transcriptome for every cell. A method was published like this back in 2017, but they delivered just one guide RNA per cell, and so they tested 71 sites, and so they were limited by power somewhat, and so they tested just this number of sites, and we were hoping to increase the power and throughput of such a setup by multiplexing the guide RNAs per cell, similar to how variants are multiplexed within a person, you could say. So that's just what we did. Guide RNAs are delivered per cell, and because each guide RNA only is likely to impact a small part of the transcriptome, you can treat each one independently when you're reassorting the population based on each guide RNA or perturbation at a time. So that's sort of our setup. We first termed this CRISPR QTL mapping as an homage to eQTL screens, but just want to note that we are aware that this is very different than actual eQTL screens, where this is a reverse genetic screen. We're inducing epigenetic perturbation because we're using CRISPR-I instead of delivering synthetic variants per cell. But to avoid nomenclature baggage, we didn't want to switch the name, so now we just refer to it as the assay formerly known as or our multiplex enhancer gene pair screen. So that's our framework, and we use this to, in a pilot experiment, study over 1,000 candidate enhancers. And we delivered 15 guide RNAs per cell and collected 47,000 single-cell RNA-seq transcriptomes. We then performed an at-scale screen that had about 4,800 new sites, in addition to some sites from the previous pilot screen included. So it's about 6,000 sites total on the screen. And we essentially then, if you count all expressed genes within a megabase for what cis enhancer gene pair tests we're testing over 78,000 at once. We delivered 28 guide RNAs per cell, and we collected over 200,000 single-cell RNA transcriptomes. So I'll tell you mostly about this at-scale screen results today. So how do we pick our candidate enhancers to test in the screen? The 4,800 sites, essentially, we required that a region in the K562 cell line, which we performed the screen in, we required the region be open chromatin, so Cas9 can easily access the underlying sequence. And then we essentially loosely picked for overlap with enhancer marks such as H3K27 acetylation, as well as transcription factor, blood-related transcription factor binding, all according to ChIP-seq. And we also required that these be outside of gene boundaries to avoid any conflation that might come from Cas9 interrupting just transcription of the gene in its gene body. So we're only testing intergenic candidate enhancers, and we delivered 2 guide RNAs per directed 2 guide RNAs for every candidate enhancer to try to account for guide RNA efficacy. So we packaged that guide RNA library into lentivirus, and then simply just pouring large amounts of virus onto cells, we achieved a high multiplicity of infection to multiplex the guide RNAs per cell. So that's how we achieved 28 guide RNAs per cell. And then we collected over 200,000 cells via the 10x genomic single-cell RNA-seq platform. And then we had packaged the guide RNAs, in a vector that was poly-A capturable based on published methods, CropSeq, et cetera, the ones mentioned here. And that allowed us to genotype simultaneously the guide RNAs in every cell just from the single cell transcriptomes alone. And to note, by multiplexing the guides, we saw perturbation of every candidate enhancer in about 900 cells, whereas if we had done a single guide RNA per cell framework, this would have required 5.8 million cells, which on its own would have been a really crazy large data set. Okay, so now we have the genotypes of the cells, and we also have their transcriptomes. So now how do we actually test, again, for potential enhancer gene pairs associations? So we simply just in silico now computationally separate out the single cell transcriptomes based on any one guide RNA or based on any one perturbed site. And then between these two in silico separated populations, we test for differential expression of any gene within 1 megabase because we wanted to limit ourselves to cis potential relationships here to avoid conflating, just to sort of simplify the chance that this is a true enhancer gene pair compared to confounders that might come from trans interactions or interchromosomal interactions. This is sort of our differential expression, our enhancer gene pair test framework. And how do we know how well it's working? Well, we included 381 control guide RNAs that target the TSS of expressed gene. So we know what genes repression should be detected in these cases. And we set a empirical FDR of about 10% of 10% based on non-targeting control guide RNAs that should have no effect in our screen. So that we set a threshold based on those. And using this, we detected 94% of TSS targeting control guide RNAs. So we're feeling pretty confident that this can detect what we're looking for here. Additionally, we detected all cells that we directed to the beta globin LCR as repressing the expected gene in infected cells. We can also detect the non-infected here. Great. Now have a look at our actual enhancer gene pair size relationship. We set the same threshold based on non-targeting guides, and we got back 664 enhancer gene pairs. The passive threshold. And to note, this is 600 enhancers that target 479 genes, and that's because candidate enhancers affect one gene. Okay, so what do these look like? We have this whole data set. What does one example look like? I'll take you through one locus. This is candidate enhancers we identified for the gene NMU, which encodes a neuropeptide neuromedan U. So if we separate our population of cells based on non-targeting control guide RNAs, and then we plot the cells just by expression of NMU, you can see non-targeting control guide RNAs should have no effect, and you see that there's no strong change in repression or no significant differential expression test returned. And then if we separate out the population of cells based off of the TSS of NMU, you can see that there is about 77% repression in the cells that do have the guide RNAs targeting the NMU TSS. And we've got a very strong differential expression test back. If we separate out the cells based on an intermediate candidate enhancer, there was no strong effect. But if we target this region that's about 90 kb upstream, there's very strong repression of effect on the order of targeting the TSS. So we termed this region E-NMU, an enhancer of NMU, and called it one of our enhancer gene pairs in our 664. So this is just one example, but now we can also, since we have 664, test for sort of genome-wide characteristics of enhancer gene pairs. So we use the Rao et al. K562 high-seed dataset from 2014 to try and look for enrichment of interaction frequency between the enhancers and TSSs of our pairs. And so for this, we're essentially ranking every pair that we detected in our screen against a distance-matched control, a set of controls within the same topologically associated domain called by that paper. And you can see in our enhancer gene pairs, there's enrichment for it being the number one, the most quote unquote interacting pair within a distance match pair within a TAD. But to note, not all of our enhancer gene pairs were the top ranked interactor. And additionally, this enrichment was not seen when shuffling either the distal locus to our TSSs or shuffling the TSS pairs to our candidate enhancer loci. And also now we can provide one of the first functionally supported distributions of the distances between enhancers and their target genes. So in this plot, I'm just going to show you the distance of enhancers, our nominated enhancers from their target genes. So if something's 250 kb upstream, it'll be marked in this region here. And we're only looking at upstream candidate enhancers to control for gene body in those distance measurements. And so here's a null distribution to start with. So this is all of our enhancers that we did pair, but instead of against this paired gene, against all expressed genes within a meg base. So this would be our null distribution. And now you can see of our or of our functionally supported enhancer gene pairs, there's a strong enrichment for being relatively close to the TSS, but it's probably median 24 kb away. So this is not all clustered close to the TSS as you may expect from CRISPR spread or something like that. But there's definitely enrichment for proximity in linear space. Additionally, now we can take a look at if the target gene is, if the candidate enhancer is always targeting its closest gene. So on this plot here, if a candidate enhancer targets its closest expressed genes, it's in the 1 column. If it's skipping a gene or targeting a second closest gene, essentially, it's in the 2 column, and so on and so forth for 3, 4, all the way to over 25 away. And about 2/3 of our candidate enhancers targeted the closest gene. So that's sort of used as a proxy in a lot of enhancer gene target measurements, and it worked about 2/3 of the time here for the candidate enhancer targeting its closest gene. So that's our data set, and I'll sort of that's all I'll take you through today, so we have enough time for discussion and questions. But in summary, we multiplexed RNAs per cell for a higher power enhancer gene pair screen. In total, between the pilot and the at-scale study, we perturbed over 80,000 potential enhancer gene pair relationships. We used this borrowed framework from the EQTL world, and we provided genome-wide characteristics of enhancer gene pairs, such as the median distance between genes and enhancers. And our work was published back in January. If you'd like more details later, and I hope future versions of this work get at why we're getting such a low return rate from the screen of 664 out of these tens of thousands that we potentially perturbed. And I'm excited for those next versions. And I'll finish with thank yous, especially to Andrew Hill, who is critical to this work, as well as the whole Shinduri lab and Jay, of course. And with that, I will take any questions.